Afternoon or morning? Okay, so your time, Yanzu. Thank you for presenting today.
Yeah, so good morning, everyone. So my name is Zhou. I'm currently a PhD student from Dr. Patrick Schnabel Lab at Iowa State University. Today, I'm going to, wait a second, sorry. So today I'm going to give you guys some update on currently the latest study from our lab on phenotyping and dissecting the genetic variation of maize azimuth canopy orientation and its impact on light interception. To begin with, we all know it's well known that the maize corn yield, the corn yield grain yield has been dramatically increased in the past 70 years. One of the major contributor to this grain yield is a better adaptation, keep breeding and selection on a better adaptation of corn plants at the higher planting density. So we are interested in what specific features that the corn plant have developed for better adaptation in the higher planting density. So to do that, we first screen a couple of commercial hybrids that planted in central Iowa as 2016. What we found out is most of the commercial hybrids that we screen exhibit, like what we show in the left figure, exhibit a specific canopy orientation pattern. So the plants, the canopy prefer to orientate towards interrow space. Therefore, they form a specific canopy pattern that a plant is perpendicular to the crop row and parallel with adjacent plant. Of course, this is not always the case. Some of the plants, like I show in the right figure, it doesn't exhibit this specific canopy orientation pattern. And this further reminds us one of the pilot studies that our lab did earlier, in which we plant 4-row B73 seedling plant in sandbench with as I show in the left figure, the upper two row, we plant the kernel specifically with the embryo facing the same direction as the planting row. And the top two row, we have the embryo facing perpendicular to the crop row, and the same direction as the crop row. And the bottom two row, we have the kernel facing perpendicular to the crop row. And then after the germinate, the lower two row, all the leaf are oriented with the same direction as the row direction. And the top two row, the leaf showing this perpendicular to the row direction. And this makes what I show in my previous slide even more interesting, because if the kernel at early stage, the leaf orientation follows the same direction as the kernel, in the field condition under which the seeds are presumably to face a random direction, what we would observe or expected to observe in the later developmental stage is a canopy shell oriented randomly instead of this the specific pattern that we orientate. So that indicates the plant potentially have some ability to form a different orientation through their developmental as the plant reaching maturity. So we did set up a simple experiment by setting up a camera on top of the plant grow in the greenhouse. And what we find that is some plants, and as I show in this video, the newly emerged leaf exhibits this ability to keep orientating. So as you can see here, so after the newly emerged leaf, they have this ability to keep readjust their leaf orientation. So this give us some hint and show that the plant actually exists. The corn plant actually have this ability to readjust their orientation, which it could explain that why the plants showing a specific canopy orientation. And when we look at the inbred, what we found out is even more diverse than what we observe in the hybrid. For example, this is 2 examples. The Mizraim 18W, in which you can see that they follow a similar canopy pattern as the commercial hybrid, with the majority of the leaf oriented towards the interrow space and parallel to each other. Meanwhile, for the Mizraim, the mode 45, what we find out is the plant, instead of pointing towards the interrow space, the leaf oriented with the same direction as the crop row. And therefore, we start to see this, the emptiness of the interrow space. So today, my talk will be divided into 2 major sections. The first one is to identify the genetic factors that contribute to this specific variation in canopy azimuth. And the second part is explore its potential role in breeding and evolution, if we got enough time left. So let's start with the first part. To begin with, we try to study the canopy orientation by simply ask whether a plant showing the same orientation pattern with the other plants in the same row. So by doing this, we can quantify different canopy orientation. For example, the first, for K45 in this picture, out of 6 plants that we planted in the crop row, 4 of them exhibit same canopy orientation pattern and parallel to each other. And for another genotype like LP5, we don't find any uniformity on the canopy orientation. Therefore, the ratio is 0. By doing this, we can quantify the canopy orientation variation. So the experiment was conducted in 2 years, in 2017 and 2018, in a diversity panel containing about 300 genotype. And the ratio was then transformed using logit transformation. In both sorry, I forgot to mention that. So in each year, the data was the parallel pattern eta ratio, eta was collected from 2 time points, 40 days after planting and 50 days after planting. So as you can see in the right figure, in both years, the 40 days after planting, the median is about 0, is at 0, which means about 50% of the plants in a row exhibits this parallel pattern. And consistently at later developmental stage, at 50 days of the planting, there is a significant decrease on the median from 0 to about minus 0.3. So that is about 30% of, so it drops from 50% to 30% of the plants that exhibit the parallel pattern. So we then look further to try to compare the consistency between the time point within each year. And interestingly, we can classify the data into 6 major clusters, with cluster 1, 6, and 4, in which they exhibit the parallel pattern fairly consistent, fairly stable. Meanwhile, for cluster 3, cluster 5, and cluster 2 on each side of the panel, they exhibit this strongly shifted parallel pattern. For example, for 40 days after planting, the bottom part at 2017, 40 days, some genotype exhibit 100%. So all the plants exhibit same canopy orientation. But at 50 days after planting, they just switch to completely random orientation. By comparing the two years data, this trait exhibit a moderate heritability at about 0.35. So we are interested by this observation that plants have showing this dramatically switched parallel pattern. So one of the hypotheses is, our first hypothesis is maybe this is because due to the light, because the plants growing bigger at 50 days after planting, so there is naturally that's the competition from adjacent plant increase, and therefore is push the plant to reorientate their canopy orientation. So in order to test that, we then collected the photosynthetic active radiation from in 2 years of 2019 and 2020 from the same diversity panel. So using this customer-built PR collecting system, so we have this. We have two liquid line sensor, liquid point sensor mounted on the top of a 6 feet tall selfie stick. This way, it enables us to capture the PAR above the canopy, so we can know that how much light is reaching the canopy. And then we mounted a liquid line sensor onto a small utility car, so we can push it through the field and deploy it to the end of the canopy to collect the ground canopy PAR. Therefore, then we subtracting the differences to estimate how much amount of light that has been intercepted by the canopy, and got this canopy reduction ratio R. So in both 2019 and 2020, from 40 days after planting to 60 days after planting, we can see that there is increasing ability, increased PAR reduction from the canopy, which exactly as we expected. We then try to compare the PAR interception reduction among different canopy orientation. So we divided based on the parallel ratio eta. We divided the canopy orientation into 3 subgroups. The first one is what we call random, in which we don't find any uniformity orientation in the genotype. And the second one is what we call off-row parallel, in which at least 50% of the plants exhibit parallel pattern and exhibit parallel to each other, and also perpendicular to the row direction. This looks exactly like what we show in the commercial hybrid. And the last one is a very specific orientation canopy pattern that we observe in our diversity panel. What we call is on-row parallel. So this pattern is the plants are still parallel with adjacent plant, but instead of have the canopy pointing towards interrow space, they pointed the same direction as the crop row. And we compare the PAR reduction in both 2019 and 2020. And what it shows is the on-row parallel consistently exhibits the lowest ability on intercepting light. Meanwhile, for random and off-row parallel, we don't see a really significant differences on the average level of PAR reduction between these two groups. So this gives us some support that different canopy orientation do show the different ability on light interception. We are then interested on what specific genetic factors that actually trigger this orientation. So we run a GWAS using parallel and PAR reduction, using both single time point data and predicted genotypic effect by combining all the time point. And we find out multiple interesting candidates. So in total, our GWAS identified 14 candidate genes, with 3 from PR identified from PR reduction, and 11 from the parallel GWAS. And when we look at the function, well, to our surprise, we find out that most of the candidate genes that we identify from parallel GWAS belong to genes that are associated with a specific function called shade warning syndrome. In case some of you are not familiar with this, shade warning syndrome is a term that summarizes the basic response to shading. And this mechanism is mainly triggered by a red light far red light ratio. As I show in the left figure, when you have shading, you basically greatly decrease the red light far red light ratio. And one of the consequences that it will trigger the plant to have more erected leaf angle and suppress bud outgrowth and petioral elongation and stem elongation. So the plants will basically look taller compared with no shading treatment. And interestingly, in corn, there's a paper published in 2003 at Plant Physiology by a group of scientists from Argentina, in which they set up a far red light filter on the south and north side of the plants. And what they find out is, compared with the black control, the plants started turning away from the direction, the south and north direction. That's after you block the far red light from this side. So this mimics exactly what we observe in our populations of the off-row parallel pattern. So our GWAS result probably give the direct evidence, the molecular evidence to what's observed in Madoni et al. In 2003 Plant Physiology paper. So encouraged by what we found, so we further conducted phenotype validation. We obtained a bunch of uniform mu mutants and grow them in 2 locations and phenotype them in 2021. So we are then interested how we can quantify this phenotypic variation. So the way we did is we try to count how many number of leaves that actually pointing towards the interrow space. So how we did is from a 4-row plot, we selected the middle 2 rows, and from each row, we consecutively selected 10 plants. Therefore, 20 plants in total from the 2-row plot, from the 2 rows. And then we roughly start from the middle of the interrow space. We roughly stretch a 24 inches wide interval. Therefore, we generate this 24 inches times 10 plants rectangle. And then from the bottom to the top, we manually counted all the leaves that entered this 24 inches wide space, this rectangle space. By doing this, we can quantify the phenotypic differences. For example, the W22, we have 63 leaf and pointing towards interrow space. And for liguleless 1, there is only about 25 leaf. So we also collect we then planted this collected this trait together with PR reduction from 15 genotype, including the wild type and the uniform mutants from the GWAS candidate, and additional several liguleless mutants that we are interested. So as the data show in the right panel, there is a strong linear correlation between the number of leaves towards interrow space and the PR reduction. What this means is you have basically you have more number of leaves pointing towards interrow space than you are expected they are going to intercept more light. And if we look at from early developmental stage to later developmental stage, as the plants reaching maturity, there is a clear linear trend that the genotype started to increase the number of leaves that pointing towards interrow space, potentially due to an increased competition from adjacent plants. And when we compare the GWAS candidate mutants, the mutants of GWAS candidate versus the wild type control, what we find out is 4 out of the 6 candidate, Ligulus 1 mu, Ligulus 2, and 2 mu mutant of phytochrom C2 exhibits this significantly reduced number of leaves towards interrow space. So this is basically confirm our GWAS result that what we are picking at is the real gene that actually contribute to the azimuth orientation. And since we also check if other, because this is single gene mutation, so we also expect other agronomic trait might be also being altered. So we collect several different agronomic trait from the GWAS, from these mutants. What we find out is some mutants, for example, the Ligulus 2, despite this mutant exhibits significant decreased number of leaves towards interrow space, but it also exhibits a higher, taller plant and also the length of and also longer leaf, which probably working together to contribute to no significant differences on the PR reduction. So what this implies is a single gene contribution to the leaf orientation, and overall contribution to the light interception might be further, might be a more complicated than what we expected, because other agronomic trait might be altered, altered or changed simultaneously. Therefore, we may not be simply so one trait. The leaf orientation might singly contribute to the general PR interception. And among these mutants, one of the Ligulus 1 is the most is the mutant that exhibits the strongest altered orientation phenotype, together with the strongest loose ability of PR reduction. Interestingly, so as you can, and when we talk about azimuth orientation, we then measure the azimuth orientation of both Ligulus 1 and W22. From what we show here, W22, the canopy orientation exhibit 2 major clusters. So the first cluster is oriented between 60 to 90 degree. Oh, sorry, I forgot to mention the 0 to 180. Here's the 0 to 180 degree is the row direction. So 90 means the leaf is pointing perpendicular to the row, and also the 270. So the 60 and 90, 30, 300 to 270 means that the W22 are mainly pointing towards interrow space. Meanwhile, for the Ligulus 1 mutants, this bimodal distribution pattern completely disappeared. And instead, we started seeing increasing number of leaves that are pointing towards the crop row direction. For example, the 30 to 330 and 150 to 210. So you can see there is a significant increased number. Meanwhile, for W22, you barely see any observation at these two angle region. And interestingly, when we look at other Ligulus mutant, we screened Ligulus 2 mutants on Ligulus 2, and another 2 mutants on Ligulus 3 another 2 mutants on Ligulus 3. In all the Ligulus mutants that we screened, the mutants compared with wild type exhibit a similar pattern with significantly altered less number of leaves that actually pointing towards growing towards an interrow space. And if you come, so this reminds us the paper that I mentioned earlier, which reported by the Madoni et al. So for when plants when they set up the far red light filter on each side of the plants, as plants started turning away, and this phenomenon mimic exactly what we should look here in A632. Meanwhile, the liguleless 2 mutants is look exactly like the black control. So by seeing this, our first reaction is maybe this is the liguleless instead of in addition to what it has been reported in the literature on the vertical leaf angle. What we should observe here, the altered azimuth orientation, is potentially due to a malfunction on how plants and sensing and respond to adjacent plants. So to test that, we planted both liguleless 2 and liguleless 1 mutant together with their wild type in 4 planting density. The smallest one is about 14,000 plants per hectare, and the highest one is about 60,000 plants per hectare. So this corresponding to acre is 7,000 plants per acre and 28,000 plants per acre. So when we first look at the lowest planting density, the mutants, according to the trait, the number of leaf towards interrow space that we counted, we barely see any differences between mutants and wild type. Meanwhile, when we gradually increase the planting density, we starting showing that the differences start getting larger and larger. And what even more interesting is liguleless 2. As you can see here, the A632 and liguleless 2, they have a completely reverse reaction to the planting density. So what we observe here is basically confirm our hypothesis that in addition to regulator ligu and oracle development, the liguleless gene may also participate in the plant's sensing adjacent plant and a better adaptation to higher planting density. So next, I would like to spend some time to talk about explore the potential role of canopy azimuth in evolution and breeding. Unfortunately, for this section, I don't have many solid data. It's mainly our hypothesis and some preliminary data that we feel like to share with the community. So speaking about this red light, far red light trigger shade warning syndrome, in current literature, what they observe is a trait that mostly happened on a vertical plane. But it never been in both Arabidopsis and tomato. No report have been showing that the plant exhibits this ability to alter their azimuth angle under shade or other light treatment. A similar report can be found in the sunflower, in which the plant exhibits this orientate or reorientate to tracking the sun direction, but it has been proved that this is through the stem. So again, this ability in sunflower is not achieved where the altered leaf orientation. Meanwhile, when we look back for other grass species, we notice that sorghum. So the red figure is a sorghum planted in northern Texas. The sorghum also exhibits this ability, the canopy preference of our leaf orientation, similar to corn, in which you can see that the majority of the leaf pointing towards interrow space. So what this figure shows is that the sorghum potentially also preserves this ability, exhibits this ability to readjust their leaf azimuth orientation according to adjacent plants. So this leads to two interesting hypotheses. The first hypothesis is this ability is shared between directly inherited from their co-ancestor between sorghum and the maize. Or it could also be due to a parallel selection, because both corn and sorghum have been intensively selected under better adaptation to high planting density. And in addition, we also tested some pioneer era hybrids in 2018. From the 40 days after planting and 50 days after planting, you can see the era hybrid at 40 days. Era hybrid in 40 days after planting, its median parallel pattern is lower than diversity panel. Meanwhile, at 50 days after planting, the parallel pattern started to increase. So there's more leaf. The era hybrid started to exhibit the parallel pattern. So that is exactly the time point when we see that that parallel and perpendicular canopy orientation in commercial hybrids. And so, and when we compare multiple inbred, what we show here is a different inbred. Actually, there is a diverse response on the leaf on how the canopy reoriented under high planting density versus low planting density. So we found several genotypes like the Mithrin 18W, A632, and CML333, in which the high planting density, we have this, there is significantly increased number of leaf pointing towards interrow space. Meanwhile, for some genotypes like MO45 and W22, at least in central Iowa, we don't see the orientation is fairly consistent across different planting density. So this implies that this function is the number of leaf pointing towards interrow space. This trait can be used to evaluate how plant to adapt to better adaptation. Better adaptation to high planting density. So to summarize, some corn plants, what we found out is some corn plants can readjust canopy orientation in response to increased planting density. And our GWAS on parallel and PR reduction identify genes related to shade warning syndrome and leaf liquid development. And this is further validated by our mutagenesis. And also we found out, in addition to gene translate light response protein, the canopy orientation may also be regulated by gene participate in liquid development. And we have some preliminary evidence indicate that this canopy azimuth pattern is observed in both maize and sorghum and potentially under breeding selection. I would like to thank my lab, especially my PI, Dr. Patrick Schnabel, for his advice and support in this study, and also Dr. Aaron Kuzmic for we have intensely collaborated on this project, and also Dr. Dan Natalieton for his advice on stats analysis, and also three Schnabel Lab alumni, Dr. Shrikant Srinivasa, Dr. Stampan Hay, and Dr. Lakshmi Atigala. With that, I would like to stop here and start taking questions.